Our next speaker is Dr. Hongjun Sung. He is the Perelman Professor of Neuroscience at the Perelman School of Medicine at the University of Pennsylvania. He received his BS from Peking University, his MA from Columbia University, and his PhD from the University of California at San Diego. And he did his postdoctoral training at the Salk Institute for Biological Sciences. He started his independent laboratory at Johns Hopkins University School of Medicine in 2002, and then moved to the University of Pennsylvania in 2017. The research in Dr. Sung's laboratory focuses on plasticity in the adult mammalian nervous system, in particular, the adult hippocampal neurogenesis and neuroepigenetics and neuroepitranscriptomics. His laboratory also applies patient-derived stem cells in 2D cultures and 3D brain organoids to model human brain development and disorders. He is also a member of the National Academy of Medicine.
Okay, great. Let me show my slide. Well, thanks, Jack, for a nice introduction. I really would like to thank the organizers to put this public lecture together, and I'm honored to invite me to give a talk to all of you today. So I'm going to tell you today about many years of study to look at demethylation in the nervous system. And we're showing here is a landscape picture. I hope at the end of my talk, I'll tell you what we try to use this landscape drawing to show. As you heard, let me see if I can move the slide. As you heard nicely from Vanessa at the introduction, in the whole body, we have exactly the same DNA, but we have many different cell types. And the way we can generate different cells from the same DNA is through a process of differentiation and epigenetic changes. And this involving a demethylation to shut off the genes. So this process, demethylation, is mediated by an enzyme called DMTs. And once you have the Cs become methylated, because the carbon-carbon bond is actually very stable. And for this reason, people believe that demethylation is very, very stable in the fully differentiated cells. Actually, for a very good reason. Because, for example, we have liver cells. We never would expect the liver cell to express gene in the neurons. And vice versa, we don't want the neurons to express liver genes. So really, there's no reason for turnaround genes during the differentiation to be shut it off. But for that reason, people believe that demethylation never change after the cell become fully differentiated. We become interested in this question from a neuroscience angle. So this actually is the project studied by my first graduate student. And we were attracted by the hypothesis put forward by Francis Crick. And this is related to human memory. What he proposed actually raised the question many years ago in a short piece in Nature. He asked the question, the time span of human memory. And often it's a matter of years, sometimes even tens of years. Yet it's believed that almost all the molecules in our body, with the exception of DNA, turns over in a matter of days, weeks, or at the most a few months. He asked the question, how then a memory is stored in the brain? So a trace of it is relatively immune to molecular turnover. And what he tried to hint to is the DNA could be the substrate for long-term memory. And Robin Holliday, later on, years later, actually proposed that specifically. He proposed that maybe the memory can be stored in DNA in a version that you have two versions of DNA. It could be the basal level, or it can be methylated. This is just like the computer, you have 0 and 1s. So you can use this store information in the DNA. Because DNA is very stable, you can have a long-term memory to be stored. So for those who actually, this is a very nice hypothesis. For many years, actually have not been tested, approved, or disapproved. But it has an intrinsic problem. So people old enough, you remember old days, we don't really have DVDs. We don't have what we have is called CDs, which you can burn a CD to record whatever information into them. The problem with CD is not reversible. When you write to them, you cannot erase it. You can keep it forever. And this is part of the DNA problem we have. If demethylation is not reversible, when you write to the DNA to store memories, you cannot change that. And the only way to do that is you have, for example, USBs nowadays we're using you can change, you can write to the USB, you can erase it. So all this hypothesis raised the question whether DNA methylation is reversible in the nervous system. So when I started my own lab, actually with my first graduate student, that's where it's a risk-taking for new graduate student, Dinko Ma. Now he's an assistant professor at UCSF now. We asked the question, can we develop a model system to answer this question, which is, how can you look at a short time, a short or transient activation of neurons, how can that lead to long-term effects? And the system he used at the time was a generation of newborn neurons in the hippocampus. And this actually was shown nicely by Jack Perrin's group at University of Michigan, where he showed that if we induce seizures in mice, so the mice or rat actually have seizures for a short period of time, but if you look at the number of new cells generated on the figure on the right, you can see nothing happens on day 1. By day 3, you can see an increase in the number of neurons generated. So by that time, you do not have any seizures anymore. And you can see this effect actually lasts for more than 13 days. So this is the case where you have a transient seizure, and after some delay, you actually have long-lasting impact. So the hypothesis Dinko at the time put it on is how can you turn the short-term seizures into long-term changes of neurogenesis, perhaps through an epigenetic mechanism, where you can change the chromatin, maybe to change the DNA, eventually have a long-lasting effect. To make the long story short, eventually, indeed, he finds this is the case. And he finds that acute activation of neural circuitries, activation of neurons, leads to a cascade of signal transductions in neurons. And what eventually leads to is decreased demethylation in neurons. What you can see on the right, the individual black dots at the individual Cs are methylated in the neurons at the basal level for many of genes. These genes, for example, BDNF or FGF, they have heavily methylated. After acute neuronal activation, what you see that these neurons reduce these black dots, that leads to deactivation, a demethylation of DNA. So this is one of the first examples to show that the demethylation neurons are not as stable as we saw. You actually can go through a process of demethylation. So this actually raises a number of questions we want to address in the following years. So the 3 questions we have, which is how dynamic is demethylation in neurons? Here we show examples for 2 genes, how widespread that is. The second question we have in mind is what is the molecular mechanism can control this process or mediate this process. And finally, we're interested to understand what are functions of DNA demethylation at the molecular, cellular, and hopefully at the behavior level. So this leads to the second graduate student, another graduate student in my lab, Junjie Guo. He's now also has his own lab now at the Yale University. He actually is going to come visit Penn to give a talk next month. So what he did to do when he was a student in the lab is took an unbiased approach to look at demethylation levels in neurons across the genome. What we find here is showing on here is we have the red showing the demethylation levels to be heavily methylated. The blue are not methylated at the basal level at 0 hour. You can see 4 hours later, you can see many of the DNA Cs change from red to blue, suggesting they become demethylated. By 24 hours, they come back again. You can see also the non-methylated Cs become methylated by 4 hours. They come back in 24 hours. What this is suggesting is there are many sites of DNAs can change their demethylation status. So this provides a view that globally, demethylation actually is very dynamic in neurons, unlike what we saw before. In the fully differentiated cells, demethylation is very stable and they never change. So this is the first surprising he found. The second one is when you hear about people talk about DNA methylation, they normally only happens in the CG context, which means the C is always before the G when the C can be methylated. When Junjie did the whole genome by sulfur sequencing to look at the methylation in the whole genome, surprisingly, he found that in neurons, they are very special. The methylation not only happens in the context of CG, it can also happen in other contexts, especially CAs. And this is important because, for example, we identified the non-CG methylation can be bound by molecules such as MECP2. The mutation of MECP2 can lead to Rett syndrome. The third surprise he helped to find is that normally we believe that demethylation block the transcription because they block the binding of transcription factors to the DNA consensus sequence. Instead, what Junjie found is that the demethylation do not block the transcription factor bind to the site, but they change their specificity. So basically, many transcription factors can bind to certain sequences, but the DNA is not methylated, but they bind to another set of sequences when the C is methylated. So DNA methylation actually produces a diversity of binding sites for transcription factors instead of blocking DNA methylation. So basically, this unbiased study leads to a number of surprises we never saw in the nervous system. So now next question is how DNA methylation is mediated. This is also Junju's work, and what he found is that DNA methylation is not a simple switch where they switch from methylated C to unmethylated C, but instead it goes through a process involving multiple steps, especially involving DNA repair. So basically, the Cs firstly become sort of oxidized, and then they go through DNA repair pathway. Then eventually they return back to C. So it's a series of biological process involving this process. It's not a single switch. So next we're asking, what is the function of this DNA demethylation? So here shows what we find that the enzymes involved in this process, the first enzyme is the TET enzyme, which oxidizes the DNA. And we find that the TET expression levels is regulated by neuronal activity. For example, if you block neuronal activity by blocking action potentials with TTX, you see decreased level of TET expression. If you block the GABA receptors, block the inhibited transmission to increase neural activity, leads to increase of TET expression. Functionally, here shows the trace where we record neurons communicate with each other through synaptic transmission. What you can see, this individual line is events of neurotransmitter release detected by the neurons. And what we find is that by blocking the TET enzyme, you see an increase of synaptic transmission become neurons, suggesting DNA methylation is fundamentally important for neuronal communication through synapse. So how about changes in neural activity? Here, neurons has actually special ability to adapt to themselves. So when the neurons at the basal level, if you suppress neural activity by blocking action potentials, they actually can increase the synapse composition to increase the level of neural activity. This is called scaling up. If the neurons are very active, they can downregulate the network activity by removing synaptic receptors at the synapse. This is called synaptic scaling down. And basically what we find is that by removing the DNA demethylation pathway, by knocking on TET3, the cells become stuck. They cannot change their synaptic level. They cannot scale up. They also cannot scale down. So what this is suggesting then, a DNA demethylation actually provides a platform for their plasticity, for flexibility. So we then perform a series of other studies to look at this at multiple levels. At the gene expression level, we know that neural activity can induce gene expression. For example, here shows you see certain genes goes up, certain genes goes down. But if we block the DNA demethylation by knocking out one key component here, GET45, you can see the neurons can still change the gene expression in response to neural activity, but the degree is reduced. So basically, they're not as robust. So they lose some type of flexibility in they change their gene expression in response to neural activity. I already showed they lose their ability to change their ability to snap communication between each other by synaptic transmission. We also behavior analysis, you heard from Erica. In this case, we perform a test where we let the mice learn the spatial environment and find a platform in the water maze. So after training, the mice become very good at finding the platform. You can see here over time, the mice actually become quicker and quicker to find this platform. So the knockout mice actually can do better. They can find this platform faster. And then we challenge the mice. Once they learn to find the platform, we move the platform to a new place and allows them to learn again. So this is reverse learning paradigm. You ask the flexibility. The mice, they relearn the new place, or instead they actually keep remembering old place. Now you can see the red dots here. The mice with a knockout, they cannot have DNA demethylation. Now they do much worse. So this suggesting this sort of lack or reduce behavior flexibility for these mice to learn new tasks or find new places. Is this also involved in some kind of disease state? And interesting, we find that, for example, if you look at the genes, the GET45 genes involving DNA methylation, they are abnormally expressed in autistic kids' brains. So here we look at postmodern brain, look at the brain expression of GET45. You can see in autistic kids, the expression level is lower, suggesting actually they may have less ability for DNA demethylation. So to test directly whether autistic spectrum disorder like behavior is affected in these mice, we perform so-called three-chamber test. So this is also very similar to Erica's test, where the mice are social animals. They like to interact with novel objects, but also they like to interact with other mice, especially novel mice. So you put the mice in the middle. Now, if you have sort of a mice instead of object, they like to spend more time with the other mice. So here it shows, you can see in the wild-type mice, they spend more time with the mice. In the knockout mice, you can see they're less interested in another mice, so they spend less time, but they still can find the mice. So now we test a so-called social novelty test. So instead of the old mice here, you put the new mice into the cage, and the old mice actually would like to explore the new mice. So they spend more time interacting with new mice instead of old mice. You can see in the wild-type mice, now they go to the new mice. But you can see in the knockout mice, they still spend the same time with old mice versus the new mice. So they lose the ability to differentiate the new versus old. So there's another level suggesting there's a lack of behavior flexibility. To summarize, I told you today, basically, we'll find that a demethylation actually is very dynamic. Activity can lead to DNA demethylation, and demethylation controls flexibility at the multiple levels. At the molecular level, it controls activity-induced late onset genes. At the cellular level, to control basal synapse transmission and synaptic scaling. I did not show you the data. They also control the surface glutamate receptor levels. At the behavior level, they control social and cognitive behavior flexibility. We also provide evidence suggesting that there may be this pathway is defective in autistic spectrum disorder. So finally, I want to go back to the picture I showed you at the beginning. So here is actually a painting drawn by my son when he was actually 13 years old. I tried to depict the idea of demethylation. For many years in the field, the dogma is that demethylation is very, very stable in fully differentiated cells, such as the neurons, they don't divide. So it's just like the mountains showing here, they're very stable, they don't change, they change about their height in 1,000 years, they're very, very stable. For a while now, people think, well, maybe there's some plasticity, some dynamics. So maybe they are like the trees in the wind, they can shake, they can move a little bit. What the new data, including our data, shows that demethylation could be very, very dynamic in response to changes in environment, for example, just like the running water showing here. I think that's it. I would like to tell you, I'll be happy to answer any questions you may have.
Thank you. I'm just going to move Jake back on screen. Oh, okay, he's already here.
Great, that was a great talk, Dr. Sung. So now we will move on to the Q&A for the talk. And again, feel free to either raise your hand and come on stage to ask your question or put your questions in the Q&A section. All right, let's see here. So I guess we'll start with this question from Ethan. Do you know whether some of the effects of DNA demethylation are specifically related to certain brain regions? Could you test this?
Oh, actually, that's actually a great question. So we know, for example, I did not mention the region we actually studied that is in the hippocampus, specifically in the dendritic hippocampus. We also show that, for example, I did not show the data here. If you put the mice in the enriched environment, as Erica showed you in his sort of cages with all different toys or others, interaction with other mice, and they show interactions, they show DNA demethylation in specific inguinal cells. Studies also have been done. For example, if we expose the mice to light and change their light, and then the visual cortex become lights up, and there are demethylation changes in the visual cortex. So all these actually are region specific, are cell type specific.
All right, thanks. Our next question is from Vanessa, who asks, are there drugs that can aid in changes of DNA methylation that can treat autistic individuals?
That's a tough question. This is actually a two-part question. The first part is, are there any drugs you can change DNA methylation? So there are drugs, as I mentioned at the beginning, the DNA methylation is mediated by the enzyme called DMTs. They are inhibitors for DNA methyltransferase. So you can block DNA methylation. So far, there's no drugs for DNA demethylation yet. So for example, TET enzyme, there's no drug yet. I think it's an interesting idea. By modulating DNA demethylation, maybe you can treat with autistic spectrum disorder. And this actually people have been trying to achieve in other ways. For example, you can change neural activity, you can change metatropic glutamate receptors, the mGLURs, and those has been tried in clinical trials in Fragile X, for example. So people are thinking about that. I try to use different ways to achieve changes in epigenetic demethylation for different disorders. And the other one would be really on the block is the Rett syndrome, which is a mutation of a protein mediated demethylation binding. Great question.
All right, great. Our next question is from Dr. Wimmer, who asks, deficits in learning flexibility are common with aging. Are there known changes in DNA methylation with aging? Is it known whether they contribute to age-related changes in cognition?
That's a great question too. So actually, there is so-called epigenetic clock. So people actually using DNA methylation as clock to ID exactly how old the cells are. So people has identified markers in DNA methylation in different cell types. I find that they correlate with the real age. So there's a cell age, there's real human age, for example. So they are DNA methylation as a marker. So basically, to answer your first question, indeed, aging is associated with changes in DNA methylation. So basically, the specific loci actually can tell you exactly how old the cell is. Whether they contribute age-related changes in function, and that's another good question. It's not definitive yet, but clearly this could be one contributing factor for decline of cognitive abilities during aging or in other aging-related disorders, for example, Alzheimer's disease or others.
All right, thank you. Our next question comes from Ashley, who asks, has DNA methylation been investigated as a factor to modulate cellular identity? It moved. Such as in protocols for direct neuron conversion or astrocyte neuron conversion?
Oh, definitely, right? I mean, the hallmark of DNA methylation has been used as a marker for cell identity. As Vanessa actually gave a good example as well, is the neurons because they have a specific DNA methylation profiles. And liver cells, for example, have a different profiles. And that's exactly how they are different, basically. And due conversion, for example, neurons convert to astrocytes or astroconverted neurons, they change the methylation status. So basically, they change the identity by changing the DNA methylation. So that is known. So basically, you have to change the DNA methylation profiles for the cell type conversion.
All right, great. And the last question we'll do now, and again, remember, if you have any more burning questions, stick around till the end to ask it at the general Q&A. But Chin asks, does inflammatory cytokines affect the methylation levels?
Well, there are many. We have not specifically looked at this. People, for example, they look at IL-6. We actually look at IL-6 as well. And those cytokines actually can change synapse transmission. If you can change synapse transmission, you will change neural activity. My guess or my expectations is they will change neural activity. So basically, the demethylation is really a substrate of many targets. Basically, it could be environment, it could be inflammation, it could be experience. So I expect that they will. And this will be cell type specific as well. Well, thanks for all the great questions.
Yeah, thank you guys for all the great questions. So now we'll move on to our next speaker, Dr. Matt Wimmer.